Hi, everyone. Today, I'm really excited to present the CIC speaker, Dr. Neil Oxtoby. Dr. Oxtoby is a neurodegenerative disease progression modeler with a PhD in theoretical quantum physics, who is currently a principal research fellow based in the computer science department at University College London in the UK. And he also plays guitar, sings, and occasionally takes selfies while wearing a colleague's lab coat. So everyone, a warm welcome. And Dr. Oxtoby, the floor is yours.
Thank you very much. It's great to be here in my living room, but I'd rather be there. Thanks for the invitation. So I'm going to tell you a little bit about what I do, and that is in the progression of neurodegenerative disease, or POND group, in the Center for Medical Image Computing at the Department of Computer Science at UCL. And I've borrowed the title for this talk from one of my recent papers, and we'll get to that in a second. So the two main points then, I'll give you a little bit of background and motivation for what I'm talking about, which is mostly Alzheimer's disease, but neurodegenerative diseases in general. I'll give a bit of background for that for those who aren't too familiar with it. The key message is that there's a lack of well-defined and consistent disease time axis for us to understand disease progression. And that's where data-driven disease progression modeling comes in, which is essentially a combination of mathematics, human insight, a bit of machine learning, and some big, although that's relative, data. The talk is based on two review papers, one that I did about 5 years after switching over from physics into neurodegenerative disease research called Imaging Plus X, and a paper that came out just last month in Nature Reviews Neuroscience that I co-first authored with Alex Young. So in that paper, we defined what a data-driven disease progression model is. The two essential elements are these two. It constructs a quantitative timeline of the disease, and it's directly informed by measured data. This data usually comes in a sort of a cohort style baseline, visit 1, visit 2, multiple biomarkers in different colors from multiple individuals. And here's a few choice individuals in bold. If you look at their biomarkers combined, you'll see that this snapshot from individual 1, just a few visits, can be even just one visit, has relatively low or normal biomarker values for these 3 colored biomarkers. Individual 2 in the middle has some moderate and high measures for some of the biomarkers. And individual M here is a little bit later on. And the idea behind data-driven disease progression modeling is to combine snapshots like this into a longer picture, a more holistic picture of the group level trajectory of disease progression, at the same time as estimating this data-driven time axis and the abnormality signature that comes along with it. So in this paper as well, we defined the disease progression models to come in 2 categories: phenomenological, where we look at the phenomena, what's going on, describe that, and fit the time axis, and pathophysiological, or also known as mechanistic models. So using my physics brain in a little paper a little while ago, I called the phenomenological models top-down models, things that you can measure. Think of if you're thinking of a brain imaging, something from the top down. And bottom up is the pathophysiology, so understanding things from underneath and then constructing a model that we can infer some parameters about that from measured data. So jumping into some background then, for those of you who might not be too familiar with Alzheimer's disease, it's a neurodegenerative condition, so we lose brain tissue, and it's defined actually by post-mortem histopathology. So when someone says that living person has Alzheimer's disease, it's sort of a bit of an approximation. And the post-mortem histopathology has this staging mechanism invented by Braak and Braak. But the clinical syndrome that we see in the hospitals, in the memory clinics, is basically a decline in memory. But it's not just memory. There are other factors as well. Alzheimer's has a long pre-symptomatic period with potentially decades of pathology going on before the symptoms kick in and before making it virtually impossible to identify future patients before those symptoms do kick in. We've learned a bit over the years. We know some risk factors, including things from genetics. And we've learned quite a bit from some more rare inherited familial forms of the disease, where we can recruit children of patients who have these mutations before they actually develop symptoms. I'll show you a little bit of an example of that. The key message is that there is heterogeneity in the Alzheimer's syndrome in the onset, in the progression speeds and the signature of progression, and the underlying pathology. But as many of you will know, in an imaging center, imaging can probe this pathology in vivo. So there is something that we can do, including MRI and PET modalities and others. The reason that we don't have any treatments, so actually I should have said the other way around, we don't understand the disease as well as we think. So how do we develop treatments? Well, there's been this amyloid cascade hypothesis around for quite some time. The pro is that there is plenty of supporting evidence. So amyloid is a misfolded protein that can clump into plaques. And okay, just distracted by a chat in the Zoom call, no worries. And a potential argument against the amyloid cascade hypothesis is that many trials for quite some time have had somewhat of a sketchy track record of showing efficacy, despite plenty of promising results in early phase trials. A lot of these anti-amyloid therapies aren't showing too much efficacy in the large phase 3 trials needed to get to market. The next natural question is why these clinical trials failing? Hundreds since 2003, a few successes recently, but again, not perfect. Could be that we're treating people too late, maybe the wrong time. We should be focusing on prevention rather than cure. There could be too much individual variability or subgroup variability between patients. We could be treating the wrong people in these trials. Could be treating them for not long enough. We could be looking at the wrong things to define what is an efficacious treatment. Currently, it's mostly clinical benefit. The patient has to see a benefit for an organization like the FDA in the US to approve a drug, rather than just show that they're normalizing biological biomarker measurements. We could even be focusing on the wrong targets. We could not understand the biology very well. Comorbidities could be causing a problem and need to be treated, or multiple targets need to be treated at the same time. So clinical trials are actually pretty, they're pretty involved. They've done quite a lot of work at trying to find the right people at the right time, starting with screening over on the left with various risk factors like age, biggest risk factor for these age-related neurodegenerative diseases. And mostly this screening takes a lot of individuals and involves mostly clinical measurements. Blood biomarkers are quite exciting these days. And then once you've screened a large number of people, filtering down to a few, slightly fewer at least, look at some exclusion criteria to remove things that might confound the trial, incidental findings, vascular pathology, and structural imaging comes in there. And then you look to screen in individuals at even higher risk. So you start paying for the more expensive scans like amyloid and tau PET scans and CSF tests, all the while reducing the number of subjects that are going to be entering your trial. And you can even stratify by the risk of decline if you have a pretty unique design of your trial. And you can use various modalities of data in order to do that. It's a couple of good papers to read on that if you're interested. But focusing on the imaging side of things, here's 3 different PET images from normal patient, a normal case on top, and a patient or an abnormal case on the bottom. So you've got amyloid PET on the left, the primary pathology of Alzheimer's disease. The heat map on the top in the normal individual is pretty cold relative to the heat map at the bottom in the abnormal individual. The tau PET doesn't appear quite as spread, dispersed as the amyloid, but it does appear as another heat map. And FDG is a tracer that measures, so I just said tau is the second pathology in Alzheimer's disease, second main one. And FDG measures glucose metabolism. So you go from a heat map to a cooler map when your metabolism is disrupted by something like Alzheimer's disease. But even these high resolution neuroimaging approaches are often just converted into some sort of binary value in order to make a decision. But this disease, Alzheimer's disease, and most neurodegenerative diseases, and actually I think probably most age-related diseases, are more complex than that. For those of you who are familiar with Alzheimer's disease would have been counting how long it took until I presented this slide. This is called the Jack curves from Cliff Jack Jr. of the Mayo Lab, who presented this hypothetical model of Alzheimer's disease progression being a sequence of biomarker changes. So these biomarkers are dynamic for a short period of time relative to the whole time course, going from left to right, and then they stabilize, they plateau. So they're no longer good for tracking disease progression. But in combination, they can be having a sequence of dynamic biomarkers. This hypothetical model was supported by sort of human evidence and anecdotes and a little bit of data, but this model itself is just a cartoon of the concept of that cascade of dynamic biomarkers. But as I said, the disease may actually have different trajectories, different cascades. So in the top left, you've got something that's a fairly common hypothesis of Alzheimer's disease, starting with vascular pathology in blue, going through amyloid and tau, the primary pathologies, into MRI and metabolism on FDG, followed by cognitive decline. On the right, you may have tau happening before amyloid, green then orange. On the bottom right, you may have a variant where tau is quite late. And on the bottom left, where vascular pathology is quite late and the Alzheimer's pathology is the primary combination there. So these are all nice hypothetical models, but if you have a hypothetical model like this or a data-driven model, you can use it. Let's say you've got a disease or a disease subtype, disease 1 labeled here on the left and disease 2 on the right, or subtype 2, and the corresponding biomarker signatures as a abnormality increasing vertically again and disease stage left to right. What can you do with this? If you have an individual with measurements of each of these biomarkers, all color-coded to match, then you line up those measurements with each of the models. And as those measurements line up with the trajectories, that increases the likelihood of that individual data, set of data points there, cross-sectional data, individual visit, single visit, being consistent with that disease. So the likelihood plotted down to the bottom here would increase. So for example, this case had blue and red biomarkers quite low and very similar to disease 1 early on, and green and orange biomarkers that were quite similar later on. But in disease 2, all the biomarkers lined up, giving an increased likelihood. And this is an example of a differential diagnosis application, which could be used for stratification in a trial or for patient management in a memory clinic. So if you have this with a quantitative model, then you can actually do something a bit more than what you can with traditional models, because traditional models have an unknown disease stage or time. You can only get so far with a hypothesis. So with that background of Alzheimer's disease and hypothetical models, I'm going to give you a little whirlwind tour of the journey to data-driven disease progression modeling. That's a little bit of a story, but it's unlike if you've ever done quantum physics in undergraduate like I did in another life, the historical flow, the timeline actually makes logical sense as well. So in the early 2000s, nearly a quarter of a century ago, traditional models equated disease stage with symptoms. So you use some kind of a clock. So in this paper from Rachel Scahill et al., looked at neuronal atrophy on T1 weighted MRI. So the heat map indicates more atrophy from pre-symptomatic through mild and moderate AD using MMSE as some sort of a clock of the disease time axis. So equating stage with symptoms, which is obviously useful in the symptomatic phase of the disease. But if you don't have that, but you do have a dominantly inherited cohort of people who have inherited a mutation and are 99% likely to develop the symptoms, then you can line them up alongside this estimated years from symptom onset, which is usually based on when their parent was first diagnosed. And you can do a cross-sectional fit of the curve of various biomarkers. On the left here, we've got amyloid deposition in a particular region of the brain. In the middle, it's the MMSE, general cognitive test. And on the right, hippocampal volume declining. And the blue line is the non-carriers, the children who are lucky enough to not inherit their mutation from their parents. And the mutation carriers are in brown/orange. So you can build a trajectory or a model of given a known disease time. Essentially, this is regression. But this is very rare, these familial forms of Alzheimer's disease, talking under 1% of cases. So enter pattern recognition towards the end of 2002 to 2008 period. Paper here with a heat map on the top row of Alzheimer's disease cases versus controls. These are just group differences. And on the bottom, Alzheimer's disease versus another dementia, frontotemporal dementia. You can do just a pattern recognition supervised machine learning classifier on MRI in your 2 case control style study. A group in Finland invented something called the disease state fingerprint, where each biomarker, so this is in this histogram here, or this density plot here, is just hippocampal volume. They convert that into a disease state index. They do that across multiple biomarkers on the left here, red indicating more like the patient cohort in the training data, and blue indicating more like the control cohort. And this, all those disease state indexes together become a disease state fingerprint, and they can convert that into a risk score. And I believe that has been commercialized. Around about the middle of this period, something called the Alzheimer's Disease Neuroimaging Initiative came along. And if you've done any research in Alzheimer's disease, you will have heard of this. It's a large study mostly in North America, multiple sites recruiting individuals and looking at the value, trying to understand the value of neuroimaging for Alzheimer's disease studies, particularly clinical trials. But that data set's free and open, available to use. And if you haven't yet used it, there are controls in there as well if you just want to do some brain mapping kind of stuff. So around about 2010, some of that evidence that emerged from the ADNI initiative, sharing their data and people analyzing it, fed into Cliff Jack's hypothetical curves, the paper that came out in 2010. And they updated it looking at some uncertainty. So there was some sort of minimum threshold, which was, you could argue what the motivation was there was for to introduce this. But as a physicist, I embrace this. I love uncertainty, and it's really important to quantify it. And then another group later on added vasculature pathology that could precede amyloid or tau. And again, another hypothetical model. But all these things started happening around about 2010. But around about the same time, computer scientists started to hear about Alzheimer's disease and started to hear about ADNI in particular. And they said, well, if there's data, then surely we can do something a little bit more data-driven. And in 2011, a pseudo-time method came out that estimates a discrete pseudo-disease time. It's called the event-based model, which essentially just estimates a sequence of progression. So there's no notion of the quantifying the time here. It's just a sort of a ranking of a sequence with some uncertainty shown in the grayscale off-diagonal elements, indicating that these events may happen simultaneously or interchangeably. That was invented by Hubert Fontaine and Danny Alexander, and generalized from familial disease to sporadic by Alex Young and others a couple of years later. The cool thing about this is like my cartoon before, you can bring patient data along and line it up with the model and stage them. So in blue, we've got cognitively normals from the ADNI study. This is the same training data that was used for this model. Mostly to the left, patients, Alzheimer's disease cases, probable Alzheimer's dementia cases diagnosed towards the right in pink, and some mild cognitive impairment, a prodromal phase, a bit more spread out. And then you have a fine-grained understanding of disease progression that is, goes above and beyond this simple symptom-based clinical categorization. There are continuous versions of this that require longitudinal data to go in. So they're latent time methods, which lines up with that initial cartoon I showed you. That's the first figure of the recent review paper. Examples include the latent time joint mix model, which is just like a mixed model, a mixed effects model with a time shift parameter allowed. That came out of Mike Donohue's lab on the west coast of the US. Some item response theory model and a more flexible trajectory version of the latent time joint mix model called the Gaussian process progression model from Marco Lorenzi's lab over here in Europe. The staging system on a continuous time scale looks a little different, but same idea. Blue is towards the cognitively normal end of the Alzheimer's clinical spectrum, and red is towards the dementia end of the syndrome spectrum. We now have a nice continuous staging measure. A little while later, in 2018, Alex Young and Danny Alexander invented this thing called subtype and stage inference, which I believe Alex has told you about before, which does the same sort of thing as pseudo-time sequencing, but then adds clustering in over the top to find multiple sequences of abnormality, multiple cascades of abnormality, which again has the patient staging mechanism, but also adds this subtyping. So we've got cognitively normal here on the left, MCI, mild cognitive impairment in the middle, and Alzheimer's disease cases. You can see that the Alzheimer's cases align more closely with the corners of this ternary plot for this 3 subtype model. But even back in cognitively normals, there are some individuals whose brain volumetry or atrophy, if we call it loosely, does line up quite well with one or another of the subtypes, suggesting that potentially we can do something before the symptoms kick in, which is very exciting. The same model was applied by others. Jake Vogel et al. Looked at tau PET and found 4 subtypes of tau PET spread, which were roughly equal in proportion, suggesting that maybe Alzheimer's, the tau deposition phase of Alzheimer's is actually 4 processes. Leon Axman from Southern California looked at amyloid and tau and sequences of those, and that's recently been published in Brain. And others have used Sustain as well for various things, including schizophrenia more recently, and actually arthritis in the knee. Check that one out if you're interested. So that's a whirlwind tour of data-driven disease progression models, but really I've only touched on the phenomenological models, the top-down models, the models where you have a biomarker or a set of biomarkers, whether that's an imaging biomarker or others, and you can plug that into some disease progression model that can give you a time axis and a disease signature. But what about disease mechanisms? Can we understand or explain these top-down observations of pathology that we get using some bottom-up models of mechanism and physiology? I've posed this as a question because it's not a solved problem, that's for sure. But around about the same time, in 2009 to 12, some hypotheses of neurodegeneration came out in a couple of key papers in Neuron from Bill Seeley and Helen Zhou came out about looking at these different hypotheses and how they would be potentially reflected in atrophy patterns. So you take a healthy connectivity graph and describe the brain's connections in terms of various hypotheses of what might cause that neurodegeneration. For example, it could be wear and tear. Regions in the brain that are more active or more highly connected could be more vulnerable to the disease. The selective vulnerability could be down to something else. And there's been recent papers looking at gene expression, for example. Use it or lose it. Brain regions that aren't very well connected. Maybe the lack of activity, lack of exercise. Maybe that is what makes you susceptible to pathology in neurodegenerative conditions. And these papers basically compared a few different hypotheses and looked at how the mathematical description would be reflected in atrophy. That was the first start into trying to understand the mechanisms of these diseases. The next step was basically trying to actually model the dynamics or the kinetics, more strictly, of the pathology spreading. So proteins like amyloid and tau, or abnormal versions of them, would spread like prions, that's a whole nother talk, through connections in the brain. So two big papers, 2012, as Ashish Raj's group came up with the network diffusion model, which is essentially a heat equation on a graph. So the pathology spreads through the brain like heat would along the connections. A couple of years later, Yasser Turi Medina and others came up with this epidemic spreading model, which is an agent-based thing. And as the name would suggest, epidemic, it's like someone who's infected walking through a bunch of people who aren't infected and then infecting them, and it spreads like that. This is a figure from that paper showing healthy controls over on the left and the increasing size of the pathology in various regions towards Alzheimer's disease, and some correlations in the predictions that the model could make, usually starting from an epicenter in the brain and spreading along the brain's structural connectome or functional connectome in more recent papers, and accumulating in a certain pattern that can then be measured using neuroimaging. Shortly after that, well, it started being developed shortly after that, a few years ago now. Some more physics-inspired models from network and spreading included things like modeling the misfolding kinetics. And here's a couple of names that will probably appeal to the mathematicians among you. There's a reaction diffusion equation, which the chemists would know about, the Fischer-Kolmogorov differential equation that describes reactions and spread of interacting or reacting agents. The heterodimer, which included normal and abnormal proteins and included the concept of clearance and production. So you could have these sort of competing processes going on underneath. And the Smolachowski equation, which is a statistical physics workhorse, which added in the size of protein aggregates to the equation. The point being, there's a lot of stuff going on under the hood. These are all essentially microscopic parameters, and ultimately the models describe how they relate to macroscopic or measurable parameters. You need to be careful with these models to make sure that they are identifiable. Sometimes you need to have some dimensional analysis in there to remove parameters. They give us clues, at least on this level, until till the imaging or other measurements get even better. We can actually measure these parameters. These models are pretty good for what they are now. For example, coming out of Alan Gorielli's lab at Oxford, in collaboration with the Biofinder study in Sweden, here's a paper currently in preprint, I believe, where they looked at spreading, local production. So this is just accumulation in a region or regions across the brain of tau pathology versus transport or spreading. So their differential equation, their spreading equation, spreads along the brain's connectivity network and has terms for production and transport. And they can fit multiple models of these to the data that they got from ADNI on the top row and BioFinder on the bottom row, and show the likelihood of these, the estimates of these parameters for three different phases. That's what the three different colors are of Alzheimer's disease, presumably. So this is amyloid negatives in blue, accumulating amyloid, but not yet tau. These are tau spreading and tau production parameters being estimated. That's amyloid positives, tau negatives in yellow. And then once tau is accumulated, amyloid positive and tau positives in green. So you can see that the local production model seems to, the parameter seems to be higher than the transport in the later phase when you have both amyloid and tau positivity. And this was consistent in the BioFinder study as well. But in the amyloid positive phase, their parameters are the other way around. So the conclusion of this paper was that spreading or transport along the brain's connectivity networks of tau pathology in Alzheimer's disease is more important earlier in the disease, and then later on local production, potentially from a prion-like epidemic kind of reinfection process, potentially, that wasn't included in the model, takes over. So it starts with transport and spreading along the connection, and later on local production is more important. So that's essentially the mechanistic models. There's spreading and appearance and a lot of involvement of brain connectivity. So there's a lot of work in there that can potentially be done on the imaging side of things as well, because as I'm sure many of you know, worked in connectivity, estimated from neuroimaging, there's a few question marks over things like tractography. So let's talk about some applications then. So back to the paper. Here's an example of a paper from 2013, which was based on a data-driven model that was only published, actually the maths of it was only published a few years after this paper. But this is on the vertical axis, we've got amyloid accumulation in amyloid PET. So this is an SUVR. And on the horizontal axis, the time estimated by the model. These colored box plots are cognitively normal MCI and Alzheimer's disease groups in amyloid positives and negatives, and those two separate boxes. And then the lines show some clinically meaningful levels. So a low level of amyloid to a high level of amyloid that might be meaningful for recruitment into a clinical trial, for example, and the time estimate of when it takes to get there. So this paper from Victor Villamagne and others was one of the first ones that gave us a quantitative estimate of the decades-long pathology underpinning Alzheimer's disease. And the sustain algorithm is applied by Jake Vogel to two tau PET data sets, different traces, found 4 subtypes with reasonable confidence of these individual dots, individual cases being subtyped with a probability close to 1, right to the corners. And each of these subtypes showed differential cognitive decline. So this is a very large data set, so only MMSE, general cognitive measure, not specific to episodic memory or executive function, which seems to be quite important in Alzheimer's disease, but still showed a differential decline. And all that was done was input was tau PET SUVRs into the sustain algorithm. A simpler version, so just the event-based model. This is from a paper myself and a former postdoc of mine, Cameron Shand, where we found the disease progression model of the cognitive measures that were included in this old trial, and all the controls are at 0, the cognitive normals. MCI is in orange, nice spread of multifactorial cognitive decline. And the Alzheimer's disease cases, again, this is the ADNI study, had more severe cognitive decline. And we found that if we split the cohort in an MCI trial, it was a trial of, donepezil and vitamin E, published in New England Journal of Medicine 2005, I believe. If we split the cohort and just included the MCIs at the end, I don't actually have the histogram of the trial data here, and just look at those with MCIs with more advanced cognitive decline, then the donepezil in blue showed an improvement. This is a survival curve. It showed an improvement up to about 12 months before it normalized. And this is known. Donepezil is a treatment that's been available for Alzheimer's disease for a couple of decades, and it's known that it improves symptoms for a couple of years maximum before the pathology takes over and patients who did improve in symptoms catch up, essentially. Their cognitive decline catches up with people who weren't treated. But in the original trial, when everyone was included, this difference didn't exist either, showing that potentially this very simple data-driven disease progression model could be useful for potentially enriching a future trial or stratifying cases by the severity of their cognitive decline. In this case, could be a biomarker profile. So, whirlwind tour, a couple of highlight applications. There's more details in the paper. Do go check it out. Quick summary. This is the thing that I'd like you to take home, is that this thing that we call data-driven disease progression modeling goes beyond these black box approaches that you can see sometimes, particularly in some of the more recent versions of machine learning, to incorporate human insight, not just human insight in neuronal network, artificial neuronal network architecture, but actual biological insight, clinical insight, to help guide the design of these models, and can leverage machine learning as well to discover patterns that aren't there, that are pre-known. I'd like you to hope that I've convinced you that this can aid disease understanding at multiple scales. It doesn't do a perfect job, but we're working on it. And it can support clinical decision making. That was one of the examples in the applications I just showed. And just a little plug to wrap up then, if you are interested in trying this stuff out, where myself and colleagues are advocates for open science and education. So we put together this resource called Disease Progression Modeling. So I did this together with Igor Koval, Sara Garbarino, Marco Lorenzi, and Vikram Venkataravan a few years ago. And this includes examples for a few different models invented by most of these guys, and some background reading as well if you're interested. It looks a little bit like this, and some of the notebooks you can even run on Google Colab if you don't have the resources to do it yourself. None of them require GPUs anyway. And something else that I did a few years ago with some of those colleagues as well, we ran a challenge, a prediction challenge. So you probably heard about Kaggle challenges if you're anywhere near data science. This wasn't a Kaggle challenge. This was a challenge we organized to predict Alzheimer's disease outcomes, and we had to get it done in a certain time, because the idea was, which was unique to this challenge, most challenges like this, they split data up and they keep some away and use that as the test data set to judge the winners of the challenge. We wanted people to predict data that didn't exist. So we did this challenge around about the time that the third phase of ADNI, the Alzheimer's Disease Neuroimaging Initiative, ADNI 3, was about to start. So they hadn't acquired any ADNI 3 data. We identified the individuals already in the ADNI study who were candidates to roll over and continue into ADNI 3. And we made entrance into this challenge predict monthly change in 3 key biomarkers for 5 years. Not to be sneezed at this as a challenge. And because of that, I think we managed to convince the Alzheimer's Society in the UK, Alzheimer's Association in the US, and Alzheimer's Research UK to chip in £30,000, British pounds, of prize money. The winners and everything, you can find all the information on the website, but you can still use that data set as a snapshot and use it as a framework for testing your predictive models, and all the results and study design are published as well. Just remains for me to say thank you to our team and the UCL Progression of Neurodegenerative Disease POND group. It's a previous grant of ours, Europond, which included a lot of those collaborators that I mentioned, and a current European consortium that we're leading called EDADS on detection of Alzheimer's subtypes even earlier. And obviously, it goes without saying, I think, but it should be said, all our wonderful collaborators across the world, the data providers, the volunteers for these studies like ADNI, all those, the patients and families in particular, and our close colleagues at the Dementia Research Centre who help to provide some of that human insight that I mentioned is such a key ingredient of data-driven disease progression modelling. Thanks.